Okay, well, it's great to have the opportunity to talk. I am very sorry that I can't be there in person, but I'm very excited to get the invitation. So I do look at targeting RNA with small molecules. Just as a little bit of background on me, I am an organic chemist, really a physical organic chemist by training, and really with a focus on molecular recognition. And so when I first approached small molecule RNA targeting, it was really from the point of asking fundamental questions about what do those interactions need to look like to drive specificity? What do the molecules need to look like? What do the RNAs need to look like? And now we've been able to apply that to modulate biological systems. Before I get started, I always like to show the faces of the people behind the science. I've been fortunate to work with an amazing group of graduate students, postdocs, and staff over the last 10 years. I know there's a lot of faces and a lot of names. Current lab members are shown in yellow, and we are always looking to grow. So this question mark should be you, could be you. But everything we do really is a collaborative effort. So I just wanted to give you a feel for the team before I talk about our motivation. So our motivation for looking at RNA, like a lot of people recently, came from this idea of the RNA revolution. So we've known for decades that DNA is transcribed into RNA, is translated into proteins, but with the ENCODE project, following the sequencing of the human genome, it became obvious that while only a very small percentage of our genome ultimately codes for proteins, the majority of it is transcribed into RNA, and a lot of this non-coding RNA can play important regulatory roles. And so when my lab first started, my goal was to better understand the role of log non-coding RNAs in metastatic cancer and whether or not we can modulate those with small molecules. Now, 10 years ago, we discovered fairly quickly that there was too much that was not known in the field about targeting RNA with small molecules and too much that was not known about linked RNA structure-function relationships to do both of those at the same time. So we actually started to look at viral regulatory RNA as model systems because there's a lot more known about their structure-function relationships, and it's a lot easier to manipulate those systems. And so while we started out just looking at this as the model system, it's actually become a really exciting part of the lab, and those are the one of the biological examples I'll talk about today. So if we think about RNA in terms of its potential as a drug target, we've made a lot of progress in targeting the bacterial ribosome with antibiotics, but as I have alluded to with the non-coding RNA, there's a huge proportion of the genome that we haven't targeted, right? So there's at least 70% we know is transcribed, the very small percentage that codes for protein, an even smaller percentage that's disease-related, and even smaller than that we can actually target, right? So less than 0.05% of our genome is actually targetable at the moment. And so the potential, if we can target even that same percentage of non-coding RNAs, or a greater percentage of mRNAs that we know belong to proteins related to disease, is enormous. And so these RNAs are important in things like cancer, cardiovascular disease, Alzheimer's. There are non-coding RNAs. I mentioned there are also mRNAs. You can control translation. You can look at splicing. There's been a lot of work in that area. But then, of course, we have RNA used by infectious agents. So viral RNA, as I alluded to, can be a great target for small molecules. And the potential here is also very promising. No one needs a reminder of how important it is to have antivirals in the current moment. And of course, bacteria and fungi also have regulatory RNAs. Right? So if you can target RNA efficiently, right, the amount of human suffering that you can relieve is almost overwhelming. But if we look at the scope of approved small molecule drugs, not surprisingly, nearly all of them target proteins. A little over 4% are thought to target DNA, and it's a very small sliver that target RNA, and all except for one are those ribosome-targeting antibiotics that I alluded to before. So things like linazolid, the aminoglycosides, the macrolides, tetracycline, all of those target ribosomal RNA. That one exception, however, is Evrysdi, or risdiplam. It's actually a derivative of the molecule shown here, but this is one where we have a nice model showing that it binds to double-stranded RNA at a specific splice site. This stabilizes an RNA protein complex, and that stabilization of this ternary complex, small molecule RNA and protein, leads to exon inclusion, producing a different protein that is a treatment for spinal muscular atrophy. And so this was really exciting to the field. Right? So it became very obvious that we can target a specific RNA that's not the ribosome in a therapeutically effective way. So we have the selectivity and efficacy to do that. But we still only have one, right? This was just approved in August of 2020. And so, moving forward, one of the things that we're thinking about is, what are the challenges or really differences in RNA targeting? And some of these are very biovisible. And so part of it is that from a molecular recognition perspective, the chemical functionality is just very limited. There are only 4 bases. It's really easy to see that from a functional group perspective, they all look really similar, certainly relative to your 20 amino acids. And even if you start to add in modifications, we're still not going to reach the diversity that you can reach with proteins. And then RNAs do tend to be more dynamic. They're not spaghetti flopping around everywhere, but they sample very discrete conformations. And each of those conformations can have a different function, which means that it's very difficult to know a priori not only what the different available conformations are for a given RNA, but which of those you would want to stabilize with your small molecule to have your desired functional outcome. All of these come together to make gaining high-resolution structures really difficult, so we can't do the structure-based drug design, or at least we can't depend on it the way that we often do for protein targeting. And once you go into a cell, the majority of RNA is protein-bound. And so if you've been optimizing just for RNA binding on its own in vitro, people often run into a lot of problems once they try to transfer that into a cell to look at function. That said, even though there are all of these challenges and differences that we need to take into account, there have been a lot of recent reports of bioactive ligands that target RNA other than the ribosome. So my lab looks at collecting this database, which I'll talk about a little bit more in a minute. But here we're just looking at your traditional drug-like small molecule. So it doesn't have aminoglycosides, peptides, oligonucleotides, just your canonical drug-like small molecules. But even if we only look at those, we can see there's been a really dramatic increase over the last 10 years. Again, really confirming that it is possible to target this RNA and have our desired biological outcome. At the same time, if you look at the total number, even if you go out to 2021, which we've now done, the total number is still less than 200. So we need ways to be more efficient at identifying functional RNA ligands. And so we approach this from 4 different angles. We do look very fundamentally at the dynamic structure and function of oncogenic non-coding RNAs. So really trying to figure out those structure-function relationships. Can they make good targets? We've also used really simple machine learning like pattern recognition to ask, what RNA structures can small molecules differentiate? What can they tell the difference between? And can we use that to choose better RNA targets? And then on the small molecule side, right, what do the good small molecules need to look like? You would imagine they're going to have a different chemical space. The biomacromolecules they're targeting have a different chemical space. But what is that? Is it specific and predictable such that we could optimize into it and build libraries around that space? And then finally, we built our own scaffold-based libraries. We started doing this about 10 years ago, before there was actually that huge increase in the reports of bioactive RNA targeting ligands. And we're still continuing this because one of the things that it allows you to do, once you figure out the synthesis, is ask really detailed atom functional group level questions about what's driving affinity and specificity in these interactions. And so we've looked at these with non-coding RNA, so this is the malabunchable helix, which I'll talk about at the end. And viral RNA is this particular one is from HIV, but I'll talk about a different example a little later. But I'm going to start with my discussion of the RNA privileged small molecule chemical state. And so I talked a little bit about our collection methods. And so once we identified all of these bioactive RNA-targeted ligands, we compared them with kind of an equivalent group from drug banks, so FDA-approved small molecules, and found that there was overlap with FDA space. So they overlap with a subset of FDA space, about 10% of that total space. But that's important because it means they satisfy your traditional medchem rules. So things that are used to predict oral availability that are still going to be really important in drug development. Of course, there are always exceptions, but the fact that we can build molecules that target and meet these existing rules is really important. But there were significant differences in structural properties. I'm just showing three example properties here, but you can see the differences in the number of nitrogens, the number of aromatic rings. We see a decrease in the number of oxygens, for example. And more importantly than these differences is the fact that the property distributions have stayed the same over time. So we've been doing this for 6 years, and those distributions have not changed. And so what that means is that every bioactive ligand that's been published in the last, actually, 17 years falls into the same distribution of chemical properties. So we feel really confident that there is something about this space. It's not scaffold space, right? So new scaffolds are discovered all the time. It's a property space. As we grow the library, one of the things that we've been able to do with this library and properties is look at whether or not we can start to further classify those, not just by what targets RNA versus proteins, but what are the different RNA substructures that we could potentially look at differentiating. And so we have found, at least preliminarily, that there are properties that can start to differentiate or that are specific to small molecules that bind bulges, for example, NG. Suggesting that eventually, once we build this library big enough, we'll be able to start to classify and predict small molecules that target certain RNA substructures, which will be even more powerful as the field moves forward. We have put all of this online in the hopes that it will be useful to the community. I won't go through all of the different capabilities. The link is there, so if you're interested at all in this space, please check it out and let us know what you think. We're not funded for it, it's a labor of love. So we would appreciate any feedback. But you can search by target, you can search by assay, you can even search by RNA secondary structure, chemical substructure. There's a lot of information in there. You can also search by nearest neighbor to see whether or not a small molecule that, say, came up in your screens or that you're planning on making is close to other bioactive RNA targeting molecules, which can tell you if you're in a good space, but also point out molecules or other RNAs that you may want to consider as controls. In our lab, we've used this to start to build our own RNA-targeted libraries. So we take those R-binding molecules that have been reported. This is just plotting them with their properties in PCA space. We first looked for commercial ligands that could satisfy this space, identified about 800. Didn't quite cover all the space. We did find binders to all of the RNAs that we've screened. This is more than 11 now, I need to update the slide, but we do identify binders, but we don't have selective binders for every RNA that we've looked at. So we decided to see where the synthetic ligands that we've been making fell onto this plot, and it turns out they cover a lot of the remaining R-bind chemical space. And if we screen these in a combined way, we now do get selective binders for every RNA that we've looked at. And actually, a lot of our active molecules come out of the synthetic libraries. So we feel really confident in this bioactive privileged RNA space and the fact that you can use it to build effective libraries. So I'll shift gears for the last part of the talk, looking at some of these scaffold-based libraries. The first example will be in the viral space. And so I'll talk about a story looking at enterovirus 71, which is the etiological agent behind hand, foot, and mouth disease. And our collaborators, Mei-Ling Lee and Gary Brewer at Rutgers, as well as Blanton Tolbert, who's now at UPenn, Philadelphia, identified StimLoop2 in the 5' ends at the beginning end of the virus as being particularly important for translation. And so this whole region is the internal ribosomal entry site, which basically means it hijacks the ribosome, the host ribosome, for viral translation. But this stem loop was particularly important, and if you make mutations to the bulge region, that significantly inhibits translation and, as a result, viral replication. So this made it a really nice target for us. We have a known structure-function relationship, and there are specific site mutations that we would like our small molecule to replicate. So we know exactly what we're looking for. And so we started by screening a subset of our amelioride library. There were several binders. We chose 6 of those based on diversity. But before optimizing for binding, we took them into biological screening. And so from that group, we identified DMA135 as having a really nice dose-dependent impact on viral-driven translation. So this is a dual luciferase assay. We have R-loop that's made all the time, just normal cap-dependent translation. And then F-loop that's only made when you have an active viral promoter. And so we have no impact on the normal translation. So we're not just generally inhibiting the ribosome or killing the cells within this 24-hour period. We are specifically inhibiting the viral-driven replication, translation. And at about the same effective concentration, we're also inhibiting viral replication, suggesting that these two mechanisms might be linked. So we were really excited about this result and wanted to probe the molecular mechanism a little bit more. And so we started by looking at known binding proteins of StimLoop2. There are several human beings known to interact. Thought that we would be displacing and activating binding proteins, so one that binds and helps recruit the ribosome. But we were completely incorrect on that hypothesis. We were stapling AQF1 binding to StimLoop2, which is repressing translation. And so we see this in cells, we also see it in vitro. And when we look by NMR, what we see is that when DMA135 binds, we go from this fairly rigid rod-like conformation, so really well-behaved in its tight Saks envelope, to a bent conformation. And that bent conformation opens up a binding site for AUF1, and that stabilizes this RNA protein complex, right? So again, just like I alluded to with risdiplam earlier, we have stabilization of a small molecule RNA protein complex, this repressed transcription, and ultimately viral replication. And I don't have time to go into all the details, but we've done follow-up studies to show that really to confirm that this is the binding site and that we're not inhibiting ADO binding to other RNAs, which is a really important biological function. But one of the things that I want to point out is that DNA-135 is not an exquisitely specific binder, right? So if we had chosen molecules based on specificity, we wouldn't have taken DNA-135 forward, right? And if we had optimized for binding, we wouldn't have seen it at all. But this was the only one that had biological activity because it was the only one that caused that conformational change. And so I think it's especially important with RNA that we move into functional assays as soon as possible because function and binding do not correlate. We see this over and over again. I mean, you want it to bind on some level, of course, right? You want target engagement. But the extent of binding and the extent of function cannot be guaranteed to correlate. So you want to do this as soon as possible so you can optimize for functional selectivity. Okay, in the very last few minutes, I'm going to switch gears kind of a lot to talk about one of those long non-coding RNAs I mentioned. So this is mallet1, and when we say long, we are talking very long. It's like 8,000 nucleotides in length, so bigger than even really your biggest mRNAs. And once it's processed, it's about 6.7, and at the 3' end, it forms this triple helix structure. And so the 3' end, which is A-rich, is wrapped around to form essentially a knot. And that knot prevents nuclease degradation. And this has been shown in several cell culture studies. This is showing the crystal structure identified by Jessica Brown when she was in the STICE lab. She's now at Notre Dame, and we're collaborating with her on this. Researchers found that if you do small mutations, in this case, even just one mutation, so you're just disrupting this one base triple, that's enough to go from a really high accumulation of mallet1 in a cell to a 75% decrease. So it's very sensitive to changes in stability. We know that overexpression is important for cancer metastasis. Like people have verified that with antisense oligonucleotides in mouse models. And so we thought this could also be a great small molecule target. Again, we have a structure-function relationship and a known specific mutation to tell us what outcome we're looking for. So I won't take you into all the backstory. We actually started with a diagonal strand scaffold that gave us stabilizers of the triple helix. We learned a lot from that, including that, again, affinity and function don't correlate. So when we looked at the diamidazine scaffold, we did a range of assays. So we looked at affinity-based assays, thermal stability, and enzymatic degradation, as well as some machine learning to see if we could predict the trends we were interested in. And with the diamidazine scaffold, we first looked for binders, every single binder, and put them into an RNase assay. So we're just seeing whether or not you added a degrading enzyme in vitro, how quickly that degrades the triple helix in the presence of a small molecule, right? So these are relative ratios, and we can see that within 5 minutes. For some of these molecules, we have very significant destabilization, so dramatic increase in degradation. And others, we see the opposite, so we see stabilization. And this gets much stronger at longer. So to be able to detect RNA on the destabilization side. And because again, confirming there's not that correlation there, and that within a single scaffold, you can see really dramatic differences in function. But, despite those differences, if we straightforward machine learning algorithm. So lasso selection of parameters and exhaustive multiple linear regression, we get reasonably predictive model for the things that are going to be degraders versus degraders. Now, we are working on adding more and more molecules to this to making it a better model, right? So there's a lot of room for improvement here, but as a kind of preliminary first pass, it shows that it will be possible to start to predict, to rationally predict molecules that will be better and better degraders. That nuclease assay, however, is gel-based, so it's hard to be super accurate, especially at a 5-minute time point. And so we have now pushed this to an RT-qPCR-based assay. And so we replaced that kind of RNase read-through with a reverse transcriptase read-through. So the idea is that if your small molecule stabilizes the triple helix, you will make less cDNA. If it destabilizes it, you'll get more cDNA, and we can pick that up by qPCR, and that's exactly what we see with examples from the literature, and with examples later from our own lab. Now, one thing to note, I kept using the air quotes when I talked about stabilization versus destabilization, and I do that because it doesn't actually make conceptual sense for a small molecule to destabilize a structure, but still have a favorable delta G of binding, right? And so what it means is that the small molecules must be stabilizing an alternate structure that's more easy to degrade. When we use the functional assays, we kind of test that directly, right? So we don't necessarily need to know what it is with functional assays. And we have been able to improve on these molecules, get tighter and tighter binding, and better and better destabilization. Right? But we do want to try to characterize this alternate conformation moving forward. So in the field, there's been a lot of, the assumption has always been that you're displacing this A tail, and that's more easy to degrade, and that's the pathway. But actually, kind of asking a lot to displace that A tail. Right? You're disrupting both Watson-Crick and hostine interactions, right, for several bases, and there are actually some alternative structures that don't require that much disruption. So we're currently, I don't have quite enough to show those results today, but we're looking into identifying what that structure could be, and if we could screen for stabilization of that structure specifically as a way to get an even better set of binders to take into our functional assays. Okay, I'm fairly certain I am we're over time. I mentioned that I worked with an amazing group of grad students and postdocs and staff. We've had a huge group of undergrads and high school students, over 30, come through the lab that have all made really important contributions. We have a number of collaborators. We're very grateful for the funding we have from a number of different agencies. And thank you again for the invitation. I'm very sorry I can't be there in person, but I will happily take any questions now or by email later.
Thanks, Amanda. There's one question, we'll take that before we...
Could you speculate about the existence of more mallet-1-like triplexes in human transcriptome?
Yeah, that's a great question. So there are very likely to be others. So there are some that we do know about. So NEAT1, for example, which is kind of closely related to mallet-1, definitely has a triplex or can form a triplex as well. The studies lab has worked on predictions that could identify other structures that are likely to form triplexes in the human transcriptome. A number of those seem really promising. They're requiring experimental follow-up, but I do think that others are likely to exist. We know that they exist in viruses, even cancer-causing viruses, for example, and that actually some pseudomonad structures have very short triplexes that can in some cases bind to similar molecules. So these definitely do exist, which means it's promising from a targeting perspective, but also something we need to think about from a selectivity perspective.
I don't think there are any more questions in the room.
I'll just have one here. Amanda, in terms of your parameters, you saw that there was some that were kind of more bulge or more kind of secondary structure. Do you think there's going to be room in, say, future to grow that? Say, if you want to kind of target like a secondary structure next to the bulge, do you think there's room, chemical space for that, or is it going to be very distinct kind of regions you're going to have to target?
So I think there will always be room, right? I mean, these are general guidelines, would say, right, that help with, optimization and building of targeted libraries. I think it'll be a very long time, if ever, before we can say that, these are the exact parameters that a bulge binding molecule needs to have. I mean, we're still regularly finding exceptions to our rules for orally available drugs, right? So I would expect there to be room, but we're not even close at the moment to knowing kind of exactly what those parameters in general would be. We can just see like some trends. Okay, they tend to have more of these properties, but we still need a lot of data to even really say that you could build toward that space.
I'd like to thank all of our speakers in this session one last time, please.